So without any further ado, I'll first introduce our first speaker, which is Colin Hoy. And he's a PhD student at the University of California, Berkeley in the lab of Robert Knight. And his talk is on multiple sequential prediction errors during reward processing in the human brain. So whenever you're ready, Colin.
Okay, great. Can you guys, let me hear actually. Looks good, and apparently you can see and hear me.
Yep.
Great, awesome. Yeah, thanks for showing up. So today I'll tell you about these multiple prediction errors that we're detecting with EEG in human subjects. And so, sorry, I'll try and kind of blow through things and give you guys the info, as much info as I can, and then maybe we can circle back to things that are poorly understood in the questions. But I wanted to start with the thank yous in case I do go over time. But obviously, my group, my PI, Bob Knight, Sheila Steiner is an undergrad that was instrumental to this work, and then the rest of the lab, as well as funding sources, Neuromatch, obviously, for hosting this amazing opportunity, Steve and Ameet for doing the back end, and then whoever's out there for paying attention. And so I'll say that this is all stuff that's under revision. So there's a bioRxiv preprint, and there's a QR code at the end of the talk, along with a GitHub repo. And that's also going to be at the end of the talk. And so I'm happy to share slides or answer questions and such. So why do we care about multiple prediction errors and reward learning? Well, obviously reward learning is pretty critical for adaptive behavior, figuring out how to get reward and avoid punishment. And so reward prediction errors, and for those who aren't familiar, that's, usually have some expectation of reward. And then the reward prediction error is whatever the difference between the actual outcome and your reward prediction was. And we know this is a quantity that's central to the reward circuits of the brain, particularly dopaminergic circuits. And it's also something really important for machine learning algorithms and reinforcement learning, which are basically one of our best ways to explain complex behavior. And then lastly, these different learning signals, the prediction errors, are also linked to very different types of clinical symptoms. And so teasing apart all of these things will help us better understand behavior, and also it'll help us sort of better understand when behavior goes wrong in disorders. So, so the background on this is that the original finding of dopamine neurons encoding reward prediction errors was kind of a big way to link reward to the brain. And then in parallel, reinforcement learning also had great developments using these reward prediction errors, particularly for stimulus outcome expectations. So you get a stimulus, and then what do you expect the reward outcome to be? And then the difference between that expectation and, again, the outcome to get a reward prediction error is really foundational to algorithms like temporal difference learning that have been able to explain a lot of behavior. But it's also a hot topic recently because new tools like optogenetics have enabled us to really dissect these dopamine circuits in the midbrain and found new pathways that are separate from the classic reward prediction error pathways that code for aversive and also non-valenced salient stimuli, so just surprising or alerting things in general. And also, reinforcement learning algorithms have really developed and incorporated a lot of these other prediction errors as well to model the world. So not just stimulus and outcome, but what is my action in between? And then what is the relationship between that action and the outcome? And so modeling all these different prediction errors, both the valence ones for reward and just the sort of surprise ones, like, I reached for the cup and I just missed, is, important for all these learning cases. And it's interesting because these are also related to some models of cognitive control that are a little closer to my work. So in parallel to the reinforcement learning in systems neuroscience, EEG has definitely been doing a lot of work on this as well. And so event-related potentials, ERPs, which Andrew's going to talk about it too, I think, are an important way to track different reward processes and learning in humans, right? Because it's non-invasive EEG. So one of the key players is the feedback-related negativity. This is an early, meaning about 200 milliseconds after you get some reward feedback, component that is central right here over the frontal midline in FCZ, which has been localized to the medial prefrontal cortex, which receives those dopaminergic reward signals. And it's localized to a theta frequency, which is 4 to 8 hertz. And really people have debated a lot about what's this mean? What is the FRN? And some key influential papers were arguing that it does track the reward prediction error and the valence of that reward signal. And there's a lot of evidence that really supports that story. But somewhere in the mid-2000s, people started to observe hey, there's an FRN after surprising but positive outcomes. So it's not just that, a negative reward prediction is eliciting this. And they were arguing that, hey, this is actually maybe a salience thing. It's not valence, it's just overall surprise. And there's been a ton of papers back and forth for multiple reasons, but some we'll get into. And so it's a little bit unclear whether it's a valenced RPE or a salience. And this is important because, the FRN is predictive of some symptoms and onsets in depression and some substance misuse disorders and addiction. So we want to figure this out. But part of the problem is that the FRN is only one of the ERPs, and it's kind of mixed in with, say, the P300, which is a famous ERP. It's a little slower, so it occurs later in 300 to 400 milliseconds, and in a different part farther back in the brain, localized to posterior PZ sensors, and is slower in the delta range. And that's related to more surprise and model updating. So also surprise and prediction errors, but not quite clear how that relates to the FRN, but also has been observed to be abnormal in disease. So teasing these things apart is kind of the goal here. And just a disclaimer, if you're an ERP aficionado, there's a lot more subtleties you can pick into separating out the FRN and the P300. And there's a tidbit in that at the end. So here are what these ERPs look like. 0, here's a feedback, and then there's the FRN, this little dip at 200 milliseconds. And here's the P300, that positivity afterwards. And the problem is, teasing apart one while not being influenced by the other one is hard. And particularly given that a lot of the traditional analysis methods are just averaging over a window, right? So if I just take the mean in that, I'm probably affected by both. And then measuring some kind of distance between peaks is also can be problematic. And so a lot of the traditional methods run into trouble separating things out. So we're going to use a regression-based approach to get around that. And so first off, our task is a timing task. So basically there's a ring of lights and you see the light go around and you're supposed to push a button when it gets back to the bottom, right? And that's ideally pushing a button at 1 second. But if you push it anywhere in that gray bar, that target zone in the tolerance, you'll get reward. And so then at feedback, you either get a green ching if you win or a red bottom if you lose. And then we have a rare sort of neutral condition that doesn't tell you if you won or lost. And so importantly, there's surprise that's both positive and negative, depending on whether it's a really hard or easy trial. So we staircase the size of this tolerance interval in order to make it really hard or really easy. And that way we control whether you're expecting to win or lose and tease apart surprise from valence. And so that means that we get about 80% correct in everybody on easy blocks, 20% correct in hard blocks. And then we're going to model this behavior by looking at the individual trial outcomes. So on the x-axis here is the tolerance, the size of that bar, which is either really small or really big. And then the sort of 1s and 0s are the individual outcomes, wins and losses. And so for every one of those trials, we can do a logistic regression across the entire data set. And that'll give us the sigmoid curve that you can convert the tolerance on any given trial, which is that Q they see the whole time, to a probability of winning. And then that basically scales to the reward function to give us an expected value, right? And that's the first part of the reward prediction error. And so then we can contrast that with the actual outcome, the 1 or the 0, and get a reward prediction error, which is the tan plot here. And now importantly, we have single trial reward prediction errors, which we can correlate with the EEG features. And of course, we can take the absolute value of that to get a non-valenced RPE magnitude, which would be like, how surprising is this reward? And based on our task design, we can also tease out a second set, a source of salience called the outcome probability, which is just how likely was this thing to appear. So there's 2 sources of salience and then the valenced reward prediction error. And what we're going to do is we're going to use that in a regression. So here's all our ERPs for all 6 outcomes in the easy, hard, win, loss, and neutral. You can see all of them have this dip for the FRN, the peak for the P3, and then some other stuff going on. And so instead of trying to average, we're going to go point by point across 50 to 500 milliseconds and do a multiple regression with all of our reward predictors and see which one corresponds to which feature. And if you look at the beta time series, so these are the model weights for those different regressors, you see that the tan RPE value, the valenced one, really peaks and has a strong effect right where that feedback related negativity is, which is some good evidence that, oh, it's right there, nailed it on the head. And then the unvalenced RPE magnitude peaks with the P300 peak. And then that second source of salience that we separated out is actually this slower thing that's even later, kind of on the downslope of the P3. And so we've got 3 different effects corresponding to 3 different ERP features. And we can use this regression approach across space too. So if we look at a tiny little window of 50 milliseconds featured on the peaks of these different regressors, we can look at where on the topography they are, and that's here. So the columns here are 3 windows locked to the peaks of those beta regressors, again, for the 3 different regressors. And you can see that in the top panel, in the early FRN window, you have this loading of RPE value peaking on the front mid sensors where we know the FRN is maximal. Whereas at the P300 peak time at 300 milliseconds, the RPE magnitude is in these posterior sensors where we know the P300 is maximal. And then we have this third salience outcome probability regressor that is even later and matches actually some frontal sensors, which is kind of an interesting effect that people haven't really teased out before. And then lastly, because again, we can apply the same regression approach across time frequency displays, we can see the frequency range of each of these effects. And again, you get RPE value in the theta time range, where we know the FRN is, RPE magnitude in the delta range, where we know the P3 is maximal, and then the probability is sort of in between, but slow and late. So we've separated these out in both time, space, and frequency by doing the behavioral modeling and the regression. And that really shifts us from trying to figure out the FRN to looking at this as an unfolding sequence of prediction errors. There's a valenced RPE with the FRN in this early mid-frontal theta. And then there's this non-valenced salient sort of how surprising was this RPE, just the magnitude of it in the P300. And then by doing this modeling approach, we've teased apart even a third component that is just tracking the outcome probability. And so that's a kind of a key takeaway here is that if you keep the high resolution in time, frequency, and space, then you can use this regression approach to separate out overlapping components, which is a really big challenge for the ERP field. So with that, I'll leave you with some future directions. So the aficionados of the FRN literature will know that there's some splitting between maybe the response for negative outcomes and punishment versus positive outcomes and reward. And so we're going to look at that because we've got a latency distinction where the FRN shifts around based on whether it's positive or negative. So we'll try and pick at that. But also we've got some intracranial data, and this is just a preliminary figure from 3 subjects showing you that the RPE value signal and the RPE magnitude signal actually don't overlap. If you look at the source of the FRN in this medial prefrontal region, they're all intermingled, but the different sites encode those different things separately. So no wonder they're mixed up on the scalp, they're mixed up in the brain. So those are the kind of future directions we'll pick at. So thanks. Anybody got questions?
Thanks so much, Colin. It was really fantastic. Yeah, if anyone has any questions, please put them in the Q&A. Yeah, I guess quickly while people are writing their questions, you talked about modeling, doing regression, which is fantastic for EG. Really gives you a lot more than just averaging across conditions. Did you have to do anything to sort of deal with the fact that your sets were unbalanced? That the amount of I forget the word, like errors you had in the hard case was very different from the easy case.
Right. Yeah, so that's definitely a problem. So that's one place where this single trial approach helps us out. Rather than sort of averaging across conditions and then having, like one ERP per condition per subject, we use a mixed effects model where we throw all the single trials in, and then we have a random effects sort of intercept for the subject. And so I think that's really crucial to this as well. It's not just using multiple regression. I'd say there's a couple pieces. There's making sure that you really model the variables of interest. So don't necessarily just rely on an ANOVA contrast or, particularly in a 2x3 design like this, the interactions get hairy. Just model them directly from the behavior. And that's the first piece. The second piece would be use the multiple regression, throw all those things in the model and let them work out which one is really tracking what at this point, and apply that at every time point, every sensor at, the typical across the topo and window of interest, and then also, across the time frequency plot. But then, that's a lot of multiple comparisons to correct for. So the last piece is doing a single trial with a mixed effects model where you can throw in all your trials, try and keep all the variance of those, but then separate out the subject factor as a random intercept. And that gives you a lot more power than doing some like subject averaging and then a group level thing as well. And so, you know, we have a pretty big data set. This actually, the nice part is, I didn't mention, this replicates across 2 cohorts. There's an initial 15 subjects where I did all the piloting, and then a 17 subject sort of replication, and all the, well, most of the effects replicate. The only things that don't replicate are those traditional measures of averaging in a window or the peak to peak measure, which says something, you know, if those traditional measures weren't going to be able to tease it apart. So yeah, having sufficient power in the design for sort of single trial, modeling the behavior, doing a mixed effects with all the subjects in single trials, and then applying that across all your dimensions is a really good way to solve this sort of overlapping component problem.
Perfect. Yeah, good to know. Great. I think we'll move on now to our second speaker,